Nobel Prize-winning doctor, just eat these every day and you will live to 100, not what you think. At this very moment, your cells are experiencing a process of aging that might be happening at an accelerated pace. Scientists possess the ability to measure this cellular aging, even observing it directly under a microscope. The protective caps at the ends of your DNA, known as telomeres, are diminishing with each passing breath, every meal consumed, and every moment of stress you endure. The profound work of 3 Nobel laureates has illuminated the underlying reasons for this phenomenon and has provided actionable strategies to mitigate its effects and foster longevity. Dr. Elizabeth Blackburn, through her groundbreaking research, identified the intricate biological clock ticking within each cell. Concurrently, Dr. Yoshinori Osumi unveiled the remarkable mechanism of autophagy, where your body diligently recycles its own damaged components to maintain youthful vitality. Further contributing to this understanding, Dr. Venki Ramakrishnan elucidated the critical role of protein factories within your cells, the ribosomes, as pivotal players in the quest for a longer, healthier life. The remarkable aspect of these discoveries is that simple, everyday foods readily available in your kitchen possess the capacity to communicate with your DNA, signaling your cells to initiate repair processes and bolster your defense against age-related diseases. Before we delve into the specific dietary recommendations championed by these eminent Nobel laureates, if this is your inaugural visit to our platform, we encourage you to subscribe. This ensures you remain consistently informed with science-backed health insights that are demonstrably effective. Furthermore, we invite you to share your location in the comments section. We genuinely cherish connecting with our global audience who share a common aspiration for a life characterized by greater health and longevity. Dr. Elizabeth Blackburn, a recipient of the 2009 Nobel Prize, revolutionized our understanding of cellular aging with her discovery of telomeres and the enzyme telomerase. To conceptualize their function, consider the analogy of shoelaces. Each end of a shoelace is protected by a plastic tip called an aglet, which prevents the lace from unraveling. Telomeres serve precisely this protective role for your DNA. Every instance your cells divide, whether to replace aging skin cells, mend wounds, or combat infections, your telomeres experience a reduction in length. When these telomeres become critically short, your cells lose their ability to function optimally, leading to cellular senescence, or aging, and ultimately, cell death. This process is a significant contributing factor to the development of debilitating conditions such as heart disease, Alzheimer's, diabetes, and cancer, the very diseases that account for the majority of human mortality. Dr. Blackburn's seminal research, meticulously documented and published in esteemed medical journals, provided compelling evidence that chronic stress accelerates telomere shortening. In a notable study conducted in collaboration with psychologist Dr. Alyssa Eppel, mothers providing care for chronically ill children exhibited telomeres that biologically appeared a decade older than their chronological age. The duration of their caregiving responsibilities and the intensity of their perceived stress were directly correlated with the observed shortening of their telomeres. However, a monumental breakthrough emerged from this research: the realization that dietary choices hold the power to safeguard your telomeres. Studies published in prestigious journals like The Lancet Oncology have demonstrated that individuals who embraced specific lifestyle modifications, crucially including the incorporation of certain foods, experienced a remarkable increase in telomere activity, ranging from 29% to an astonishing 84% within a mere 3 months. Dr. Dean Ornish collaborated with Dr. Blackburn on this pivotal research. A subsequent 5-year follow-up study yielded even more remarkable results. Participants adhering to these lifestyle changes observed a 10% increase in telomere length, while the control group, who did not implement these changes, experienced a 3% shortening of their telomeres. The foods that exhibit this protective effect on telomeres share a common, potent characteristic. They are exceptionally rich in antioxidants and omega-3 fatty acids. Research Research emanating from Ohio State University, published in Brain Behavior and Immunity, established a direct correlation between higher omega-3 levels and longer telomeres. Furthermore, investigations conducted at the University of California, San Francisco, UCSF, where Dr. Blackburn is affiliated, tracked 608 patients diagnosed with heart disease over a 5-year period. The findings revealed that individuals with the highest blood levels of omega-3 fatty acids displayed the slowest rate of telomere shortening. This significant study was published in the Journal of the American Medical Association. Additional research appearing in Clinical Nutrition indicated that omega-3 fatty acids present in tissues can effectively reduce telomere attrition and actively counteract premature aging. A large-scale clinical trial known as VITAL also provided evidence suggesting that vitamin D supplementation could mitigate telomere shortening by approximately 140 base pairs over a 4-year duration. To harness these benefits, incorporating specific foods into your diet is paramount. Wild-caught salmon, sardines, and mackerel are excellent sources of omega-3s, as are walnuts and flax seeds. Blueberries, strawberries, and blackberries are replete with anthocyanins, powerful compounds that contribute to reducing DNA damage. Leafy greens such as spinach and kale, along with broccoli and vibrantly colored bell peppers, deliver essential vitamins like vitamin C, vitamin E, and folate, all of which are indispensable for robust DNA protection. Whole grains, including oats, quinoa, and brown rice, and legumes like lentils and chickpeas, provide crucial fiber and B vitamins. Scientific studies have underscored the necessity of folate for DNA synthesis, repair, and metabolic processes within cells. Elevated levels of homocysteine, a marker that folate helps to regulate, have been linked to shorter telomere lengths. Research published in the European Journal of Nutrition has indicated that individuals who consume carotenoid-rich foods, the natural pigments that impart vibrant colors to vegetables and fruits, exhibit significantly longer telomeres. Mushrooms also warrant particular attention, as vitamin D levels have been associated with telomere length, according to a 2017 study published in the Journal of Nutrition. Dr. Yoshinori Osumi, a Nobel laureate in 2016, made a profound discovery regarding autophagy, a Greek term meaning self-eating. This remarkable process involves your cells actively consuming their own damaged components as a survival and renewal mechanism. Dr. Osumi's initial investigations in the early 1990s focused on yeast cells. By subjecting them to nutrient deprivation and genetically modifying them to impair their ability to fully digest cellular material, he observed under the microscope the accumulation of tiny recycling vesicles, termed autophagosomes, within the cells. This pivotal discovery, which opened up an entirely new field of scientific inquiry, proved that autophagy is not a random event, but rather a triggered survival response. It is initiated by nutrient scarcity, prompting cells to recycle old proteins and damaged organelles for energy. This intricate process is fundamental to longevity, as it effectively prevents the accumulation of cellular debris that contributes to aging, inflammation, and disease. While autophagy was first observed in the 1960s, its underlying mechanisms remained largely enigmatic until Dr. Osumi's work in the 1990s. His research identified the specific genes responsible for autophagy in yeast and confirmed that analogous mechanisms are present in human cells, fundamentally transforming our comprehension of cellular health. Peer-reviewed scientific literature consistently demonstrates that fasting for periods of 12 to 24 hours effectively triggers autophagy. Scientists have further elucidated that calorie restriction activates genes that promote cellular preservation, rendering cells remarkably resilient to disease and various forms of cellular stress. A comprehensive study published in Science Direct in 2023 examined healthy young males observing daily fasting periods of 17 to 19 hours throughout the month of Ramadan. Blood samples analyzed during this period revealed a significant increase in autophagy-related genes, with peak activity observed around the midpoint of their fasting intervals, thus confirming the role of intermittent fasting in activating cellular cleanup processes. Intriguingly, studies originating from blue zones, regions globally renowned for their exceptionally high concentrations of centenarians, have unveiled consistent dietary and lifestyle patterns. In Ikaria, Greece, for instance, residents observe approximately 150 days of religious fasting annually. This practice, coupled with their adherence to a Mediterranean diet rich in vegetables and olive oil, is believed to contribute significantly to their remarkable longevity. Exercise also plays a crucial role in activating autophagy. Research substantiates that moderate aerobic activity and endurance training induce autophagy in muscle and brain tissues. Studies conducted on mice have documented increased autophagy in response to treadmill exercise, and emerging human data suggests that exercise can regulate autophagy in a tissue-specific manner. Furthermore, autophagy has been demonstrably linked to protective effects against neurodegenerative diseases such as Parkinson's and Alzheimer's. To actively engage this potent cellular cleanup process, consider implementing the following strategies. Time-restricted eating, where you confine your food consumption to an 8 to 10 hour window, is highly effective. For example, consuming all meals between noon and 8 cellular p.m. provides your body with a 16-hour period without food, allowing autophagy to commence its work. Curtailing constant snacking and allowing 4 to 5 hours between meals is also beneficial. Autophagy is initiated only after the depletion of glycogen stores in the liver, a process that typically takes 14 to 16 hours. During these fasting periods, it is permissible to consume water, black coffee, or green tea, as these beverages will not interrupt the fasting state. Certain foods, when consumed during your eating windows, can further support autophagy. Green tea, for instance, contains epigallocatechin gallate, EGCG, a compound known to promote autophagy. Cruciferous vegetables, including broccoli, Brussels sprouts, and cauliflower, are rich in sulforaphane, a potent activator of autophagy pathways. The key to maximizing the benefits of autophagy lies in consistency. Your body requires regular periods of fasting to effectively engage this vital cellular cleanup system. Dr. Venki Ramakrishnan, a co-recipient of the 2009 Nobel Prize in Chemistry, alongside Thomas Stites and Ada Yonath, was instrumental in revealing the atomic structure of ribosomes. These minuscule powerhouses within every cell are responsible for synthesizing all the proteins your body requires. Ribosomes function by translating genetic instructions encoded in DNA into proteins, which are the fundamental building blocks for tissues, the orchestrators of immune responses, the agents of repair, and the facilitators of countless life-sustaining processes. Every single protein in your body is meticulously crafted by ribosomes. Consequently, healthy ribosomes are essential for your cells to maintain efficient protein production, which in turn supports tissue strength, bolsters your immune system, and significantly slows down age-related decline. Dr. Ramakrishnan employed X-ray crystallography to meticulously map the ribosome's structure at an atomic level. Research published in Nature Communications has shown that safeguarding ribosome function through nutritional interventions can effectively extend both lifespan and health span. To support your ribosomes, prioritize the intake of high-quality proteins, which provide the essential amino acids they require for synthesis. Wild-caught fish, pasture-raised, eggs, and grass-fed meat consumed in moderation are excellent sources, as are plant-based proteins like hemp seeds and spirulina, which offer complete amino acid profiles. It is important to note that quantity is often less critical than quality in protein consumption. Studies indicate that a moderate protein intake, approximately 0.8 to 1 gram per kilogram of body weight, effectively supports ribosome function without placing an undue burden on cellular processes. Consuming excessive amounts of protein, conversely, can be counterproductive. Magnesium is another critical mineral for optimal ribosome function. Pumpkin seeds, almonds, spinach, and dark chocolate, with a cocoa content of 70% or higher, are superb sources. Just a quarter cup of pumpkin seeds can provide nearly half of your daily magnesium requirements. Foods rich in B vitamins are also vital for supporting ribosomal production. Leafy greens, avocados, and legumes are excellent sources of folate and other B vitamins essential for the synthesis of ribosomal RNA. Research investigating caloric restriction has yielded promising results. A study published in Nature Communications demonstrated that moderate calorie reduction in mice led to a lifespan extension that was directly proportional to the degree of restriction. This mechanism is partly attributed to a reduced metabolic burden associated with ribosome production, allowing cells to divert resources towards repair rather than continuous growth. Adequate sleep is paramount for overall health and cellular function, including ribosome activity. Research published in Nature Neuroscience has highlighted that sleep deprivation in older adults can disrupt ribosomal activity. Your cells engage in vital repair processes during sleep, including those involving ribosomes. Aiming for 7 to 8 hours of quality sleep per night is therefore crucial. Physical activity, particularly resistance training, enhances the efficiency of protein synthesis. Studies suggest that resistance training in older adults stimulates ribosome biogenesis and improves muscle protein synthesis, thereby playing a key role in maintaining muscle mass as you age. Notably, the dietary choices and lifestyle habits that support ribosome function align remarkably with the principles derived from the other Nobel Prize discoveries. Nutrient-dense diets, sufficient sleep, regular physical activity, and mindful eating all contribute synergistically to maintaining the efficient operation of your protein factories for decades. To put these principles into practice, consider a daily eating plan grounded in Nobel Prize-winning science. For your first meal, opt for 2 eggs accompanied by sautéed spinach and tomatoes, a small handful of blueberries, and a cup of green tea. For your midday meal, prepare a substantial salad featuring mixed greens, an assortment of colorful vegetables, chickpeas, and pumpkin seeds, dressed with olive oil and lemon. A side of quinoa or brown rice can complement this meal. For your evening meal, aim for wild-caught salmon or other fatty fish 2 to 3 times per week, accompanied by steamed broccoli and Brussels sprouts, and a serving of sweet potato or lentils. If you experience hunger between meals, healthy options include walnuts and berries, dark chocolate, 70% or higher cocoa content, or sliced vegetables served with hummus. The timing of your meals is also critically important. Aim to conclude your eating by 8 p.m. and refrain from eating again until noon the following day. This 16-hour fasting window is instrumental in activating autophagy and providing your cells with essential time for repair, rather than constantly processing food. During your fasting periods, prioritize hydration with water, black coffee, or green tea. It is crucial to understand that these are not magical foods. Rather, they serve as vital fuel for the microscopic machinery that sustains your life. They play a role in protecting your DNA caps, activating your body's innate cellular cleanup system, and maintaining the optimal function of your protein factories. To further enhance these benefits, integrate stress management techniques such as meditation or deep breathing exercises for at least 12 minutes daily. Regular physical activity, like walking or cycling for 30 minutes most days of the week, supports all 3 of these fundamental systems. Prioritizing 7 to 8 hours of quality sleep each night is also non-negotiable. Furthermore, cultivating and maintaining strong social connections has been shown in numerous studies to be linked to longer telomeres. Ultimately, achieving longevity is not contingent upon expensive treatments or elusive miracle supplements. The cumulative insights from 3 Nobel laureates definitively prove that your body is already equipped with sophisticated systems for self-healing, efficient cellular cleanup, and age deceleration. Dr. Blackburn's research underscored the power of diet and lifestyle in protecting telomeres, thereby preventing the onset of the most prevalent life-threatening diseases. Dr. Osumi's work revealed that consistent periods of fasting activate autophagy, your body's natural cellular recycling mechanism. Dr. Ramakrishnan's demonstrations highlighted the importance of nutrient-rich foods in supporting ribosomes, ensuring the efficient operation of your protein factories. The extensive body of research spanning over 2 decades consistently implicates telomere shortening, diminished autophagy, and declining ribosome function as key contributors to the development of cardiovascular disease, Alzheimer's, cancer, and diabetes. These are not mere theories. They represent Nobel Prize-winning science robustly supported by thousands of peer-reviewed studies. Every meal presents a profound opportunity, an opportunity to send powerful signals to your cells, encouraging them to repair, protect, and remain resilient. The most encouraging aspect of this information is that perfect adherence is not required. Begin by simply incorporating more colorful vegetables into your meals. Experiment with time-restricted eating for a few days a week. Prioritize fatty fish consumption twice weekly. If this information has provided you with a clear and actionable path toward healthier aging, please share this video with someone you care about, a parent, spouse, or friend who also desires to live a longer, healthier life. If you are ready to begin actively supporting your body's inherent longevity systems, type yes in the comments section. The choices you make today will undoubtedly be appreciated by your future self.